new faculty members in exercise science. I'm just going to let you take it away.
Thanks, Liz. Thanks for the opportunity to come here and talk to you guys today. As I was putting this together, I realized this is the first talk I've given with my new affiliation here in the Kinesiology department at Kentucky. So it's actually kind of exciting for me and exciting to move on to the next phase. So, and I'm also glad to be able to do it in the home department too. So what am I going to be talking about today? So I'm going to be talking about interventions for attenuating age-associated arterial stiffening. So what is arterial stiffening? Well, arterial stiffening is becoming it is a novel and now it's an emerging risk factor for cardiovascular disease and cardiovascular risk, as well as all, overall mortality. So it's a very important risk factor to be studying. And then we're going to kind of transition from arterial stiffness and interventions, so interventions that may reduce this arterial stiffening, specifically in the context of aging, to how this perivascular fat, or how this fat surrounding arteries may be a novel mechanism. So why do we need to study cardiovascular diseases? Well, over time, since about 1900, we've seen that deaths due to cardiovascular diseases have slowly but continually increased over this time. Only recently has it shown a slight decrease in deaths due to cardiovascular disease. Despite that, cardiovascular diseases are still the #1 killer in the United States, as well as other modern societies around the world. Cancer's been increasing, but still, cardiovascular diseases are still the #1 cause of death. So how does aging fit into this big picture? What we see here, we look at percent of the population that have some sort of cardiovascular disease. We see individuals 60 and older, so 75% of those people 60 and older that have some sort of cardiovascular disease. This is important, especially currently, because the baby boomers are now hitting about this age, 60 plus. So this is a big biomedical problem, and so we need to find ways to basically reduce the risk for cardiovascular disease and deaths. So what explains this risk for cardiovascular disease in these older individuals? Well, here are the conventional risk factors. We have just being a male increases your risk, hereditary, smoking, high blood cholesterol, blood pressure, et cetera, et cetera. These are all things you've seen. But if you notice, it only accounts for about 50% of the risk. So there's got to be something else that helps explain this and maybe can contribute to this risk. Well, one of those, obviously, is large elastic artery stiffness or arterial stiffening. So when I refer to large elastic artery stiffness or arterial stiffening, I will specifically be talking about the aorta artery as well as the carotids. And a lot of this stiffening is as a result of structural changes within the artery itself. So when I say structural, I mean you get increases in collagen, you get increases of cross-linking of proteins, which all contribute to stiffening. So how do we measure arterial stiffness? It's actually quite simple. Put one Doppler flow probe on the carotid artery, and you put a second on the femoral artery. And from this, you get two pulse waves, one at the carotid and one at the femoral. What you can do from these, you can calculate how fast the pulse wave travels down the aorta. The idea being, the faster the wave travels down the aorta, the stiffer the artery, so it's less distensible. The slower the wave, the less stiff the artery, so it's actually more distensible. So you could use the analogy of a PVC pipe, where the water would travel faster through the pipe versus a garden hose, where it would travel slower because the garden hose gives. So what happens with aging and arterial stiffness? On this population study, both in men and women, we see that as you increase in age, you see increases in pulse wave velocity. Basically what this is showing us is that at the population level, we see that individuals of older age have stiffer arteries. What does this really mean, other than older people have stiffer arteries? Well, this study really puts it in context. And what they did, they followed subjects over 8 years, and they looked at the probability of major cardiovascular events. And when you divide it out in quartiles, we see that the individuals in the highest quartile, so they had the higher pulse wave velocity, they had the stiffer arteries, were a much higher risk for having a major cardiovascular event. So this is heart attack, stroke, things along those lines. Not only that, this was independent of other risk factors, high cholesterol, high blood pressure. So all those I mentioned earlier, This is independent. So this now is starting to explain maybe a little bit of that other 50% risk. So now that we kind of see the problem, we see cardiovascular disease, maybe a big problem. We see that older individuals, there's becoming more of them, and they have a stiffer artery, and that this increases in stiffness does lead to increases in clinical problems such as heart attack and stroke. So what are the mechanisms? So at advancing age, we have increases in arterial stiffness. A lot of this is due to increases in oxidative stress and inflammation, which essentially feed off each other. From there, we have extracellular matrix alterations. So we have collagen accumulation, increases in collagen within the arteries, stiff arteries. We have reductions in elastin. This is elastic protein. So with aging, we see reductions in elastin, which then in turn stiffens the artery. We see greater cross-linking. So we have advanced glycation end products, also referred to as ages. And so what this is, you have more cross-linking of proteins. Think of it this way. You have a chain link fence, you have less cross-links. The chain link fence will give more. You have more cross-links, it won't give as much. Think about that in the context of the artery as well. And then finally, we have calcification. And the more I learn about calcification, a little calcification goes a long ways when we talk about arterial stiffness. However, that's just kind of the large overview of the mechanisms. I'm going to back off a little bit and be a little bit more focused. We're talking about how advancing age leads to oxidative stress, specifically excessive superoxide within arteries. That will lead to increases in collagen as well as increases in this cross-linking with ages, ultimately to be associated with arterial stiffness. I just want to take a moment to review our vascular anatomy because we're going to be talking a lot about the vascular anatomy. So here's our lumen, and this is where the blood flows. We have the innermost part of the artery, which is referred to as the intima. This would be your endothelial layer, followed by the next layer. This is the smooth muscle layer, also referred to the medial layer. And we have the outermost layer of the artery, the adventitia. For our purposes today, I want you to think of the medial and the adventitial layers. These outer two layers is where the ages will form. So these are non-specific, they will form within any layer, but for our purposes today, we're just going to primarily refer to them as the media and adventitia. And when we talk about collagen, I want you to think that the collagen will be in the adventitia or the outermost layer of the artery. So why do we go over that? Because in order to study arterial stiffness in humans, you would have to, and get at mechanisms or structurals, you'd have to get a piece of aorta. Quite honestly, that's pretty difficult. I don't know if any volunteers will give me a piece of aorta. I know I wouldn't, so I'm not going to ask anybody else to. So then we move to a mouse model of arterial stiffness and aging. So can we do the same measures in mice? So can we measure pulse wave velocity in mice? We certainly can. And so here's our basic setup. So here's a cartoon of it. So we put one Doppler flow probe here on the arch of the aorta, and the second on the abdominal aorta. And from that, we can calculate how fast pulse wave travels down the aorta. The faster it goes, the stiffer the artery. And here's just a real view of how we have done it in the past. The mouse is on a heated board. One Doppler flow probe here on the upper chest on the aorta, and one on the abdominal aorta. So this is non-invasive. The mouse just is asleep. They wake up, and we can do this over and over. So back to our working hypothesis, working model. So we have advancing age, least oxidative stress, particularly excessive superoxide. So we want to test, see if this actually contributes to the stiffening process. So we're going to manipulate the system and treat with an antioxidant compound called Timbal. So how does this work? With normal physiology, and everybody in this room, normal physiology is we have pro-oxidant enzymes, NADPH oxidases, and they produce superoxide. This superoxide is important for normal physiological function. From there, we have an antioxidant enzyme, or a class of them, superoxide dismutases, which takes superoxide, makes hydrogen peroxide, and eventually you make water and oxygen. However, with aging, we see increase in pro-oxidant enzyme expression and activity, which in turn leads to increase in superoxide, oxidative stress. But we also see reductions in the antioxidants. So what we did, we gave we actually gave the mimetic of these antioxidants to mice to see if we could reduce the superoxide and in turn improve arterial function. Basic protocol. So we had young mice. So all the mice labeled young will be 6 to 8 months today, and old will be 26 to 28. To strain a mouse, we gave Timpal this antioxidant in drinking water. It was a short 3-week treatment. So first thing, did our treatment actually reduce superoxide within the aortas of these mice? So when we look at superoxide production, we see it is greater in the aortas of the old compared to the young. And we give Timpal, an antioxidant. It had no effect in the young Timpal treated. However, the old Timpal is significantly reduced compared to the controls. So we did effectively reduce superoxide or the oxidative stress within the arteries. What about arterial function? So this is the pulse rate velocity or arterial stiffness measures. What we see is with aging, we see an increase in pulse rate velocity, so the arteries are getting stiffer compared to the youngs. When we give Timpal, there's no mathematical difference between young Timpal and young control. However, we give old mice Timpal, we see a significant reduction in pulse rate compared to the young controls. So we show when we reduce superoxide, we also reduce arterial stiffness, showing that oxidative stress does play a role in the stiffening process with aging. Keep moving down, look at some of the downstream mechanisms. So collagen 1. Increases in collagen lead to increased stiffness. So this is a histological segment, cross-section of the mouse aortas, and this is the brown staining is staining for collagen 1. So the darker the brown, essentially the more the collagen there is. This is going to be the only images I show of this, but just to give you an idea of how we analyze the data, collect the data and analyze it. So these arrows are pointing to the outermost layer of the adventitia. I said, remember the adventitia and collagen. And what we see here in the old controls, we have more brown staining of type 1 collagen in the olds compared to the youngs. When we treat with Timpal, the young animals have about the same as the controls. However, the old Timpal, they have less compared to the controls, showing that Timpal reduces it. But we actually quantify this to actually, so it's not qualitative. And what we see is collagen 1 increases with age and Timpal reduces it. So collagen 1 is mediated, or appears to be mediated by oxidative stress that leads to stiffening. So we decided to take this one step further and move to cell culture. So does superoxide itself actually contribute to collagen production in these cells? So the adventitia, they're called fibroblasts. So we cultured fibroblasts from a young animal. And we treated with a compound called pyrogalol. And this is a superoxide generator. So excessive superoxide, we think, leads to collagen deposition and stiffening. So when we look at collagen expression in these cells, we see that our superoxide generator increases collagen compared to the controls. So we see that superoxide does contribute. We treat with the antioxidants, so this, in this case this is the SOD, superoxide dismutase. We actually prevent the effect. So we directly show that superoxide contributes to collagen, and then we can actually block it by adding the antioxidant. So that's just in young cells, but the next question is, can you actually reverse this process? We saw in vivo in the aortas, yeah, it's possible. But we did here, we took cells, these fibroblasts from a young animal, fibroblasts from an old animal, and we cultured them. And what we see is the same thing we saw with our histology. We see that there's more collagen in these cells, old cells, compared to the young. So can we reverse this process? By giving an antioxidant SOD, can we reverse it? And indeed, we were able to reverse that effect. So showing again, not only is it sufficient for superoxide to generate collagen expression, but we can actually give the antioxidant to reverse this collagen expression in old cells. So what about ages? We talked about crossing. Well, in this study, in old mice, ages abundance was increased throughout the artery, but it was unaltered with Timpal treatment. So I need to caveat this for a couple of things. So one, it was a short study. So it was a 3-week study. And the data I didn't show. When we look at the data, it's actually beginning to go down. The ages are beginning to go down. So in my opinion, I actually think if you give a longer treatment, the ages would go down. This literature strongly suggests oxidative stress is playing a role in ages. So despite this negative finding, I still think oxidative stress plays a role in ages. I just don't have data to show you today. So back to our working model, see base and age leads to excessive superoxide. When we add Timpol, we remove this excessive superoxide. We also reduce collagen. So right now, I'm just going to shade this out. I can't say it reduces ages, but in my opinion, it will reduce ages. That ultimately is associated with reductions in arterial stiffening. So from this, we see that, yes, oxidative stress, specifically excessive superoxide, does play a role in arterial stiffness with age. So what about interventions? So we're kinesiologists, we study exercise. And in my opinion, biased as it is, exercise is one of the most effective modalities as an intervention for reducing cardiovascular risk. I think the literature would support that. And so the question is, by reducing cardiovascular risk, does it also reduce arterial stiffness, which could be the mechanism for the reduced risk? So this study done by my former postdoc mentor, long before I arrived, they did a study on that. And they looked at the beta stiffness index. So they basically, instead of looking at the aorta, they were looking at the carotid stiffness. And basically, how to interpret this, the higher the number, the stiffer the carotid is. So this was the setting period. They have a young group, a middle-aged group, and an older group. What we see is the middle-aged and older group have higher beta stiffness index, which means they have stiffer arteries. Then what they did, they look at a recreationally active group of young, middle-aged, and older adults. And what you see is basically there's no change in arterial stiffness or carotid stiffness in these recreationally active individuals. So recreationally active, 3 days a week, moderate intensity. However, when you look at endurance-trained individuals, so these are individuals 5 days a week, moderate to probably vigorous, we see that the older individuals have reduced stiffness compared to the recreationally old and the sedentary old. So yes, the question is yes, or the answer is yes, exercise can reduce arterial stiffness and potentially reduce cardiovascular risk. But this is a more endurance-trained individuals that have probably been doing this for years. So the next question they tried to answer was exercise as an intervention. So you just train older adults and reduce arterial stiffening. Short answer is yes. So what they did was a 12-week intervention program in older adults. We see that the beta stiffness is about 3, which is what it was in the other round. And after this about 12, 13-week intervention, we see that beta stiffness is decreased, so stiffness is reduced. So yes, exercise as an intervention can reduce arterial stiffness. So what's the problem between the two? These individuals exercise 5 days a week at about 75% heart rate. So they are a little more intensely exercised compared to the recreational that you saw in the previous graph. So I believe exercise reduces arterial stiffness, it just needs to be a little bit higher intensity. So why am I showing you this? Mouse stuff, and then there's that. Well, we want to know the mechanism by which this happens. So we have our older adults that voluntarily walk and either on a treadmill or outside. So we did the same for our mice. We let them voluntarily run on a wheel, and they will. The young mice will run up to 10 kilometers a night, the old about 1 kilometer a night. So they will run for you. So again, back to our basic working model. We're hypothesizing that exercise will reduce oxidative stress and our collagen ages, and ultimately arterial stiffness. So again, we look at our pulse wave velocity data, and we see the same age-related increase. And then we train these animals, again, about a 12-week training period. We see that stiffness is reduced in the old trained animals compared to old controls. Now, we don't have direct superoxide measures in these animals right now, so we looked at a marker of oxidative stress. This is nitrotyrosine, so greater nitrotyrosine abundance, the more oxidative stress is thought to occur within the artery. So what we see with the old animals, we see much more nitrotyrosine abundance compared to the young. And when you train them, the old animals have significantly reduced nitrotyrosine or oxidative stress compared to the old animals. So again, exercise reduces stiffness and reduces oxidative stress. So what about one of our structural proteins, collagen 1? So this is just the adventitial, the outermost layer of the artery. We see with aging, collagen increases, and then with training, has a profound effect. It reverses it. These are a little bit more preliminary data. This is kind of a work in progress. These are ages, or they're cross-linking. And so, although not very compelling right now, this is a small n, we see that aging increases ages, and then training tends to reverse that. So I have data where ages do go up with aging. This is very preliminary and a small n. So we're just continuing to work on this. So right now it looks like collagen, or exercise reverses collagen as well as ages across the knee. So to summarize that, exercise reduces oxidative stress, nitrotyrosine abundance. It also appears to be reducing collagen as well as ages, and ultimately leading to reductions in stiffness. So exercise, like I said, I think it's a great tool to use to reduce cardiovascular risk. However, there are some potential issues with exercise. Exercise is not for everyone. Some people it's contraindicated for, so some of the elderly, they should exercise. The other issue is some elderly shouldn't exercise, they can't exercise. So this moves us on to the next line of research, nutraceuticals. What are nutraceuticals? Other than a fancy little word. It's a food or parts of food that provide medical or health benefits, including the prevention and treatment of disease. Alternative definition is a nutrient or food believed to have curative properties. A food used as a drug. So one that we're interested in is nitrites. So why do I have a bunch of hot dogs up there? Because nitrites typically are thought to be associated with curing of meat and to be like a very bad thing. They've been associated with higher incidences of cancer and that sort of thing. However, I think that literature base is pretty weak. I think what's coming out now is nitrates. Nitrates are very important. It can be cardioprotective. And I don't know, can't speak to the cancer literature, but they seem to be very beneficial to your health. They are found naturally in foods, vegetables, carrots, beets, beetroot juice, green leafies. So as I said, they're found in foods, and they also have antioxidant properties. So it fits right within what we're interested in. So we actually did a study with actually sodium nitrite, and our working hypothesis is that sodium nitrite will reduce this oxidative stress, the superoxide, while reducing collagen and ages, and ultimately arterial stiffening. So when we look at our arterial stiffness measures for pulse wave velocity, we see the age-related increase, and this is reversed by sodium nitrite supplementation for 3 weeks. So we see that it appears to be cardioprotective, reducing stiffness. Not only that, but it does reduce superoxide. The age-related increase is superoxide in the arteries. So the old animals have much less superoxide compared to the olds. So we reduce arterial stiffness and the oxidative stress or superoxide within the artery. So moving on to our downstream part is ages. So we see the age-related increase in ages. So I promised you I do have data on that. And that nitrite actually reverses this phenomenon. However, we look at collagen. We do not see any effects of sodium nitrite on reducing the age-related increase in collagen. We see that collagen is still elevated in the olds compared to the young groups. This is finding data. We see all these associations. So now we wanted to actually get more mechanistic cause and effect data. Do ages actually contribute to arterial stiffness, and does sodium nitrite prevent that? So we decided to move ex vivo. So we took arteries out of animals and studied them. So we did. We took a young mouse. We took the aorta out, actually removed it, with the idea that we're going to do a cell culture, tissue culture experiment. So we chopped up the artery, put it in the tissue bath, and then we treated the artery. So we treated with biologically active agents for 72 hours, with or without sodium nitrite. And with that, we did ex vivo mechanical testing of arterial segments. So we just took the artery, put it on 2 wires, and we stretched it. And from that, we can determine arterial stiffness. Trust me, we had a lot of help from engineers on this. So it was a lot of work, but I think we're getting some pretty good data off of these. So when we have our elastic modulus. For our purposes, it's just the measure of arterial stiffness in our ex vivo arterial segments. So when we treat young arteries with ages, so this is just conjugated to BSA, we see that ages increases the modulus or the stiffness compared to the controls. And when we pre-treat with sodium nitrite, it actually reduces arterial stiffness. And two, when you treat with sodium nitrite, it actually prevents the detrimental effects of ages. So to summarize, so with advancing age, so we see that sodium nitrite reduces this excessive superoxide. We cannot say that collagen is part of this, but we can say we see it increases ages, and sodium nitrite blocks that. And we take it a step further, we directly show that ages alters arterial stiffness ex vivo, that is blocked by sodium nitrite. So just kind of summarize this part of the talk. So very simply, excessive superoxide production contributes to the age-associated arterial stiffness that is attenuated by both exercise and sodium nitrite. So our interventions seem to be effective at targeting superoxide and reducing stiffness. So this is labeled future directions, but it's kind of like what's current and kind of what's coming type thing. And so what are we doing now? So this gets into the kind of the adipose tissue talk, or the fat talk. And periadventitial adipose tissue plays a critical role in vascular remodeling. So everything we've looked at, collagen ages, it's vascular remodeling. And so what this study basically is showing is how this perivascular adventitious or perivascular fat actually plays a role in remodeling. So I'll give you an idea, well, does fat play a role in our remodeling and ultimately stiffness stage? So let's back up and talk about perivascular adipose tissue. I refer to it as PVAT from here on out, or PVAT. So back to our artery, we have the innermost layer, the endothelium, followed by the smooth muscle layer, and then the outermost, the adventitious. And then we have the perivascular fat here, and with most disease states, these are macrophages that infiltrate the fat, which then causes the reaction, so you actually release inflammatory molecules. This is just a few of them, IL-8, ECD1, relatase. Don't worry about those, as well as adipokines, so basically molecules that are released by the adipose, adiponectin, leptin. Importantly, ROS, reactive oxygen species, so getting at oxidative stress, we need to remember that, as well as angiotensin 2. So it seems to be playing a big role, or it could directly affect the arteries based on its adjacent location to the artery. So quite simply, I have a very simple hypothesis. Advancing age leads to perivascular oxidative stress, so excessive superoxide. That'll lead to arterial stiffness. And to test this hypothesis, we're going to go back to the standby Timbal. If you remember, Timbal reduced arterial stiffness in the old mice. So first thing we did, we looked at superoxide production or bioavailability within the fat itself. And what we see, these are again, pretty preliminary data. But with aging, we see increases in superoxide in the olds compared to the young. When we treat with Timbal, it's underpowered right now, but we see a trend for reduction in superoxide within the perivascular fat. But the question is, so we see this oxidative stress, superoxide is there, but does this PVAT play a role to arterial stiffening? To do this, we took a few different approaches, one being an in vivo approach, which I'm going to discuss now. It was more of a surgical approach. So we took our mouse, and we actually opened the mouse up, and we exposed the abdominal aorta just below the kidneys here. And what we did, we just essentially excluded the endogenous fat over, and what we did, we added exogenous fat to the animal. We sewed them back up, and they lived for another 8 weeks. To get a little bit more into the study's design, what we did is we took a young mouse, and when this animal was euthanized, we took the artery out, removed the fat from that artery. And we put it into what I refer to as a recipient mouse. We put it right onto the artery, the abdominal aorta, just as I explained. Not only did we do it with young, we did that with old, an old donor mouse. This guy was euthanized, and then he was put in, his fat was put into a young recipient mouse. And we also did this with our old Timbal mice, and again, his fat was put into a young recipient mouse. With the hypothesis being, the mouse that received the fat from another young mouse, there would be no changes in stiffness. The mouse that received the fat from the old mouse would have increases in stiffness. And this mouse that received fat from the old Timbal treated mouse would not see increases in stiffness, as shown before. So So I'm going to walk you through these data. So here's our baseline data, and we have data at 8 weeks. So this is pre-post. So these are the animals that we're going to receive the fat. This is before any surgical procedure. And this is the young animals. These are the old animals, or the animals, young animals that we're going to receive the old fat. And these are the young animals that we're going to receive the old fat that is treated. There's no change at baseline. Then we actually look 8 weeks later at the animals that received, the young animals that received the young fat. There's no change. However, when we put the old fat into the young animal, we see an increase in arterial stiffness. And again, this was prevented with Timbal. So we put the old fat with Timbal treatment into the young animal, we don't see the increase. Really getting at how oxidative stress in the fat plays a role in arterial stiffening. So there's a few problems with this. So we just dump fat into the mouths, and the question is this have a direct effect on the artery, or is something being released systemically that's causing an effect? Well, it's a good first line in vivo example of this. So, oh, excuse me, I forgot we even have these data. Sorry, these are new data. I just got them this week. But so we took the arteries out and we did our ex vivo stiffness data. So nothing fancy. We just removed the arteries. And what we see is the same trend as our in vivo data. We have two measures of stiffness. This is stiffness, this is modulus. We see that the old animals have increased stiffness. The Timbal do not. Same thing with our modulus calculations. Same thing, essentially, just two different calculations. So sorry, forgot those data. Those are new as of this week. So basically that supports our in vivo data. So to test this ex vivo, to get more direct evidence for the direct role of perivascular fat on the artery, we decided to do another culture experiment. This time, so we put our arteries in the dish. We took arteries from young and old mice, and we cultured them with or without the fat on the artery. And we also cultured them with or without Timbal, the antioxidant, for 72 hours. Then we did mechanical properties. So as I go through the graph, I'll explain a little bit more detail exactly what we did. So here's our stiffness. So here's our elastic modulus. So here's the black bars will be our young arteries, and the gray bar would be the arteries from the old animals. So what we did, we cultured these arteries for 72 hours with the fat on. Right before testing, we cut the fat off. The idea is that the fat would be secreting some sort of factor that would affect the arterial mechanics. And indeed, it did that. The old animals have a higher modulus, or greater stiffness compared to the young animals. When we removed the fat, cultured it for 72 hours, then did our mechanics, we see that there's no effect in the young animals. However, in the old, we see a significant reduction in the modulus compared to the ones that had the fat, suggesting that the fat is secreting something that's leading to stiffness. So then, does oxidative stress play a role in that? So we left the fat on, we added Timbal, and with Timbal addition, we see that the oxidative stress is removed, and it looks just like the artery that didn't have the fat. That's significantly different from the artery that did have the fat. And just as a control, we had cultured arteries without the fat with the Timbal. There's basically no change. So to summarize, advancing age leads to oxidative stress or superoxide in this fat, and when we treat with Timbal, we remove that and we reduce the arterial stiffness. Conclusions: exercise and sodium nitrite attenuate both age-associated arterial stiffening and superoxide bioavailability. This excessive PVAT superoxide production contributes to the age-associated arterial stiffness. We provide direct evidence for that. Therefore, novel translational interventions target reducing this excessive PVAT superoxide with aging should be explored further. So with that, I need to acknowledge several people from my postdoctoral lab. So I give a lot of credit and thanks to my mentor, Doug Seals. If I just take a little bit of what he taught me, I think I'll be okay. Also, I'd like to acknowledge a fellow postdoc, Schindler. She's a great colleague, great to work with, as well as a few students. Jason, he's put a lot of, a master's student, he put a lot of force and effort into developing these mechanical test. He's very engineer-minded and worked well with engineers. He kind of bridged me, physiologist, with the engineers, which was incredibly helpful. Undergrad Bryant Pham, he's incredible. I've never seen an undergrad work so hard. As well as Jackson Cooler, another undergrad, who is also fantastic and works extremely hard. So with that, I'll be happy to take any questions.
So what is Tempol? What is it?
So you have the superoxide dismutase. It basically is a mimetic of those. So you're basically adding the endogenous, or exogenously, you're adding an antioxidant enzyme that's already in.
Was this taken orally?
Yep.
You provide it in water? So
Yeah, just put water. They drink it, and
there's no side effects or anything like that?
no.
This is something humans could consume, what you're saying?
Theoretically, yes, we're considering that. Yes, it's being a consideration, strong consideration. It's used in clinical trials for, like, right now, topical treatment for cancer and that sort of thing, to remove the oxidative stress, hopefully slow down the cancer.
When you used exercises, the intervention, was it solely aerobic activity?
It was, so all we did was we put a little running wheel in their cage, and they just had free access to that. They could run as much or little as they wanted to.
And then was that the same for the human subjects as well, or were they?
They were trained. It was more guided through like 75% heart rate max, that sort of thing. So it was much more guided.
Okay.
But those mice do like to run, and they do run a fair amount. 10K a night for young and 1K for an old. Yeah. More than a lot of people do. So, yeah, like.
How does exercise decrease oxidative stress then?
Yeah, so something I didn't show, so it actually reduces some of the pro-oxidant enzymes, so like P67, reduces those as well as increasing the antioxidant enzymes and inside, some of the sides that I talked about. So some of the same enzymes that Tempol mimics. So kind of it works both ways. So it reduces the pro-oxidant as well as increasing the antioxidant. Get that balance.
Do you know, like, are there any studies that look at weight training versus aerobic exercise in their intervention for both?
There have been, and so in humans, when you resistance train, they actually won't see improvements in arterial stiffness, and sometimes it'll even get worse. However, if you couple resistance training with aerobic exercise, actually will still see improvements in reductions in stiffness. So, something about aerobic exercise. And there's only a few studies with resistance training, so I wouldn't totally write it off as not beneficial, but from what I've seen, there aren't too many beneficial studies with resistance.
How similar are the arterial properties of mice and men?
Good question.
Mice and men. I didn't plan that at all.
That's a very good question. So, between humans and any basically animal, maybe excluding monkeys, they're different. So as we referred to earlier, there's an intima on the arteries. Most rodents, pigs, whatever, dogs don't have an intimal layer. Humans do. So that's one big major difference between humans' arteries. Other than that, the basic structure is very similar, same type of cells, that sort of thing. So one thing I didn't touch on today is the intima does play a role in arterial stiffness with humans. And so that is a big difference. You just can't get around it. And I don't think any animal will, maybe monkeys, but Jason?
We talked about nitrates.
Mm-hmm.
So you're telling me I can be baking sausage for breakfast?
So I wouldn't recommend that per se, because there's some other issues with that. I would stick to your green leafies, stick your spinach, your beets, that sort of thing. So I think it's just the higher concentration, and I don't, to be honest with you, I don't know what the difference is in formulation. I mean, maybe it's just much higher concentration of nitrites in those cured beets. And there's also a lot of other bad things associated with those foods.
I guess a comment, it could be the way that you're taking it as well. A lot of these substances work in conjunction with others, don't they?
Correct.
So it might be, yeah, that you're just taking a concentrated dose of nitrates in the meat, but in the vegetables it's working with something else.
Right. There's actually a lot of clinical trials going on with nitrite. The lab I came from, they actually have one going on and it appears to be improving function, I think. So it's a big hot area these days. If you put anything in a pill so we can make money, you're going to be happy. So people are scouring the nutraceuticals, so to speak. Any questions? Yeah, Mike.
So I'll focus one an interesting thought. And one of your first slides showed that there was a slight decrease in cardiovascular disease. And I don't know whether you know this, but is there any idea like why? Like are we starting to get a hold on this? Because it seems as though health is getting worse in terms of obesity and.
So, in my opinion, I think it's going to be a transient decrease. So I think what happened is we're getting smarter as people. We're figuring out how to combat the disease process. But with that said, I think it's getting a lot more complex. So there's going to be a lot more people with cardiovascular disease. So this goes from both ends. There's going to be a lot more older people with cardiovascular disease that may present with different signs and different pathologies. But on the other hand, too, there's a lot of younger people that are overweight, obese, that are going to be presenting with a totally different type of pathology. Well, it'll still be vascular disease and cardiovascular disease, but it's going to be a little different. And so, honestly, I think the deaths are going to go back up, in my opinion, unless something's done, like everybody starts working out or quit eating what they eat, totally change to fruit and veggies. I think it will get worse.